Insects did not evolve smaller versions of vertebrate eyes. They evolved an entirely different biological philosophy, where vertebrate vision concentrates information into a single optical axis. Insect vision distributes perception across thousands of independent sensory units, each functioning as a complete micro-eye. At the cellular level, each ommatidium is a miniature visual organ. Tightly packed photoreceptor cells form rhabdomeres, surrounded by pigment cells that isolate light with astonishing precision. Histologically, insects do not have a layered retina. Instead, these rhabdomeres are engineered for speed, not clarity. Photon capture is prioritized over image resolution, evolution's radical trade-off. Molecularly, opsins are tuned for extreme kinetics. They activate and deactivate in milliseconds, allowing insects to perceive motion before it fully occurs. A fly does not react fast. It perceives earlier than we even define events. Dragonflies push this to the extreme. Specialized neurons fire only for prey-sized objects, filtering background motion entirely. The sky, trees, shadows, irrelevant. Only trajectories matter. Bees took a different path. Their photoreceptors detect ultraviolet light, revealing patterns on flowers invisible to humans. Plants co-evolved to communicate via these signals, a true interspecies chemical-light dialogue. Some eyes break even these rules. Mantis shrimp rotate each eye independently, splitting visual tasks, polarization, color, motion. No central cortex, no integration bottleneck, no delay. This is vision engineered by evolution, not aesthetics. A solution to reality itself. Insects navigate worlds far more complex than humans imagine. A tiny ant can find its nest over hundreds of meters with only chemical cues. This is not luck. It is a biochemical GPS written in pheromones and integrated by distributed neural clusters. The antennae are not simple feelers. They are microchemical laboratories, scanning molecules, detecting gradients, even minute differences in humidity. At the cellular level, neurons within the antennal lobe are highly specialized, mapping chemical space with astonishing fidelity. Each odor receptor neuron connects to a glomerulus, a functional unit that encodes precise chemical identity. Some insects integrate visual landmarks with chemical maps. Compound eyes detect polarized light, encoding sun position even through forest canopy. Molecular photoreceptors signal azimuthal orientation to central processing units. In moths, mechanosensory hairs detect wing turbulence, feeding rapid feedback into flight control neurons. A single spike in these circuits can adjust trajectory midair. Navigation is therefore not centralized. Distributed microcircuits continuously merge chemical, visual, and mechanosensory inputs. The brain is a network optimized for survival, not for reflection. Bees perform a remarkable feat, the waggle dance. They translate spatial information into temporal motion sequences, a language encoded in movement and vibration. Neurons in their mushroom bodies process these patterns, forming a memory of hive relative coordinates. Even at the molecular level, intracellular calcium dynamics within these neurons govern spike timing. The precision is such that a bee can travel kilometers, locate a nectar source, and communicate it with millimeter accuracy. Some desert ants use polarized skylight as a compass. Others rely on panoramic visual snapshots combined with proprioceptive cues. Every strategy reflects a unique evolutionary solution to orientation. This is navigation reduced to physics, chemistry, and neural computation, a feat no human-engineered system has fully replicated. Insects walk, jump, cling, and fly, yet they have no bones in their legs. Instead, evolution built a lightweight, hollow, and astonishingly strong hydraulic system. The exoskeleton forms rigid segments joined by articulated membranes, engineered to store elastic energy. Muscles contract inside, fluid pressure extends segments, a natural hydraulic actuation unseen in vertebrates. Each foot ends in claws and adhesive pads, molecularly optimized for maximum friction with minimal material. Special seedy, microscopic hairs exploit van der Waals forces, allowing a beetle to climb smooth glass effortlessly. At the histological level, muscle fibers are organized to maximize speed and endurance. Thin fibers generate rapid movements. Thick fibers provide sustained force. No vertebrate skeleton can match this combination of speed, power, and miniaturization. Joint design is remarkable. Each leg has multiple axes of rotation, arranged in a geometric hierarchy, allowing precise control of every step. The nervous system coordinates hundreds of joints simultaneously, a microengineering marvel. In grasshoppers, hind legs store elastic energy in resonant paths, like compressed springs. A single jump releases this energy explosively, propelling the insect hundreds of times its body length. Spider legs illustrate another principle. Hydraulic extension plus flexor muscles allow delicate web construction and gravity-defying climbs. The combination of pressure control and sensory feedback is unmatched. Even at the molecular scale, resilient proteins function like natural springs, returning nearly 97% of stored energy. This efficiency is evolution's answer to the physical constraints of miniaturized life. Evolution did not simply copy vertebrate limbs. It reimagined locomotion with hollow structures, hydraulic actuation, elastic storage, and molecular adhesion. Every foot, every joint, every fiber is a testament to millions of years of precise, survival-driven design.